The topic is digestion. In the previous lesson on digestion, I have explained the definition and meaning of digestion, types of digestion, intracellular and extracellular digestion, and the human digestive system, and the various stages in the process of digestion, and digestion of food nutrients like carbohydrates, proteins, and lipids in detail. So this lesson is the continuation of the earlier lesson on digestion. With the help of this lesson, the learners will be able to know and understand the definition and meaning of digestion, mechanism of absorption, assimilation, digestion of cellulose, and role of gastrointestinal hormones in digestion. The digested food materials are to be supplied to all cells of the body for the production of energy, growth, and metabolism. This is possible through two important processes called absorption and assimilation. In the process of absorption, the end products of the digestion enter into the epithelium of the intestinal wall, and from there they reach into blood capillaries and lymph vessels. The capillaries unite to form a hepatic portal vein, and this vein enters into the liver. From the liver, these end products they enter into the main circulation through hepatic veins. In the process of assimilation, the end products of digestion are supplied to, they are transported through blood circulation and supplied to different tissues of the body, and they are utilized for various metabolic activities. Absorption is a process by which the end products of digestion, that is glucose, amino acids, fats, fatty acids and glycerol, they pass through intestinal mucosa into the blood or lymph. The absorbed substances finally reach the tissues which utilize them for their activities. This process is called as assimilation. In order to enter into the bloodstream, the end products of digestion have to cross two linings. One is epithelial lining of the intestine and endothelial lining of the capillaries. Very few substances are absorbed in the mouth. Some drugs and alcohol, they are absorbed in the mouth and stomach, whereas large amount of substances are absorbed in the small intestine. About 90% of the nutrients are absorbed in the small intestine. The inner layer of the intestine has a finger-like projections. They are called as villi. So these villi, they increase the area of absorption, the surface area of absorption. And they have some brush border. These villi will have brush border, and these brush border, they open into the lumen of the intestine. So they are called as microvilli. The microvilli will enhance the rate of absorption. In this figure, we can see these are the finger-like projections, and these are called as villi. These villi, they increase the surface area of absorption. And these villi will have appendages at their end. These are called as microvilli. At the end of these villi, we'll see the microvilli. Microvilli is commonly called as brush border. The epithelial cells will have the villi at their terminal end. This image shows the movement of absorbed nutrients in the blood and lymph. So this is the villus. This is the villus which is present in the intestinal wall. So this villus has blood capillaries. These are the blood capillaries, and each villus consists of the blood capillary as well as a small vessel of lymphatic vessel called as lacteal. The monosaccharides and amino acids are absorbed by the blood capillaries. Fatty acids and glycerol are absorbed by the lacteals. So these absorbed nutrients, they enter into the hepatic portal vein which enters into the liver. The capillaries which are present in the villus, they unite together and they enter into a hepatic portal vein which enters into the liver, and from the liver the nutrients containing blood will flow into the main circulation. While the lacteals, they unite to form a lymphatic vessels, and this lymphatic vessel, it will enter into the main circulation near the left subclavian duct. Now let us see about mechanism of absorption. The digested food materials are absorbed by different methods. One is passive diffusion, osmosis, active transport, and facilitated diffusion. In this slide, we can see the absorption of carbohydrates in the epithelial cells of small intestine. The end products of carbohydrate digestion are monosaccharides, namely glucose, galactose, and fructose. So among these, the glucose and galactose, they are transported into the cell from the lumen of the intestine by secondary active transport, which is energy dependent. That is, it requires energy to enter into the epithelial cells. And these molecules require sodium ion. A symporter carrier will transport the glucose along with the sodium ions into the epithelial cell. The sodium ion concentration is always kept low in the cell by the basolateral region by diffusing the sodium ions out at the expense of energy in the presence of sodium potassium ATPase enzyme. As the concentration of the glucose molecules increases in the cell, they are diffused into the bloodstream. This is the blood. They are diffused into the bloodstream through facilitated diffusion, glucose and galactose. And the fructose ions are also absorbed into the epithelial cell, and from there to the blood via facilitated diffusion. So all these monosaccharides, first glucose and galactose are transported along with sodium ions by secondary active transport. Fructose is transported by facilitated diffusion. From the base of the cell, they are transported into the blood through facilitated diffusion. Some pentose sugars are absorbed by simple diffusion. Now let us see about the absorption of amino acids. The end products of protein digestion are amino acids some few dipeptides and tripeptides also remain. Amino acids are transported by secondary active transport with the energy consumption, that is energy dependent secondary active transport. In the same manner the glucose is transported. So here we can see this is the lumen of intestine. These are the small amino acids. These amino acids, they enter into the epithelial cell along with the sodium ions, secondary active transport along with sodium ions, and the sodium ions they are thrown out with the help of the sodium potassium ATPase enzyme from the cell, and the amino acids later on from the base of the cell, they enter into the capillaries by facilitated diffusion. And the dipeptides and the tripeptides which remain, they are transported by a carrier molecule into the blood capillaries. Now let us see about absorption of lipids. The end products of lipid digestion are monoglycerides and fatty acids. Monoglycerides and fatty acids or absorption is a very complicated process. They are not absorbed in their original form. They undergo modification and transform into distinct forms called micelles and chylomicrons. In the first phase, the monoglycerides and fatty acids associate with the bile salts, cholesterol, phospholipids and fat-soluble vitamins to form into droplets called micelles. These micelles, they enter into the enterocytes by fatty acid transporter or by diffusion. In the enterocyte, the micelles are transported into the endoplasmic reticulum and are resynthesized into triglycerides. The triglycerides, along with cholesterol and phospholipids, they together form into a group covered by a lipoprotein layer. So this distinct form is called as chylomicron. So chylomicrons are designed for the transportation of lipids in the circulation. So these chylomicrons are released from the enterocyte into the lacteal present in the villi by a process called exocytosis. So these lacteals, they enter from the lacteals, the chylomicrons enter into lymphatic vessel, and from lymphatic vessel the chylomicrons they reach the large veins which are present in the neck region, and from there again they enter into the blood circulation. Once again the triglycerides and the lipoproteins present in the chylomicrons are hydrolyzed by an enzyme called lipoprotein lipase which is present in the endothelial wall of the capillaries. Then fatty acids and monoglycerols are formed, and these are transported, they are diffused into the cells. Now let us see the absorption of other substances. Water is absorbed in almost all the regions of the alimentary canal. Water is absorbed in the mouth, stomach, small intestine and large intestine. Most absorption of the water occurs in the large intestine. Vitamin B and C, which are water soluble vitamins, they are absorbed along with water. Fat-soluble vitamins are absorbed in the small intestine along with monoglycerols and fatty acids in the presence of bile salts. In this image, we can see the absorption of end products of digestion. Here, this row shows the different end products of digestion which are present in the lumen of the intestine, and this is the mode of transport, and this is the epithelial cell, and from the epithelial cell they are transported to blood. So glucose and galactose, they are transported by secondary active transport along with sodium ions, and from here the epithelial cells to the blood through facilitated diffusion. Here we can see fructose completely by facilitated diffusion. And the amino acids are transported by active transport, that is secondary active transport along with sodium ions, and from here by diffusion into the blood. And dipeptides and tripeptides, they are also transported by secondary active transport but along with H plus ions. And coming to the lipids, short chain fatty acids, they are transported by simple diffusion and from here to the outside, that is the blood, from here from the cell to the blood. And coming to the lipids, they transform into micellase. They transform into micellase along with some other molecules like that is cholesterol, phospholipids, fat-soluble vitamins, and they enter into the enterocyte, and in the enterocyte in the triglyceride they form into triglycerides, and later on they form into a distinct another form called chylomicron, and these chylomicrons are released from the enterocyte into lacteal of villus. Now let us see about the assimilation of food. The absorbed food consists of monosaccharides, that is glucose, amino acids and lipids. They enter into the blood circulation and are utilized by the cells for various metabolic activities. Glucose is oxidized in the process of cellular respiration and energy is released. This energy is utilized for various metabolic activities. The excess amount of glucose that is present is transformed into glycogen. Glycogen is the reserve food material, and as and when required, this glycogen is again broken down to glucose. So glycogen is stored in the liver as well as in muscle tissue. The amino acids are utilized in the synthesis of many enzymes and proteins which are essential from time to time for various metabolic activities. And the fatty acids and glycerol, some of these fatty acids and glycerol, they are used in the synthesis of plasma membrane, and the excess of these are stored in the adipose tissue beneath the subdermal layer. Now let us see about digestion of cellulose. Herbivores feed on plants and plant products like fruits, vegetables, leaves and grains. Plant foods consist of cellulose, hemicellulose and pectin. Cellulose is a polysaccharide The glucose units in cellulose are linked by beta-1,4-glycosidic bonds. The polysaccharide digestive enzymes like salivary amylase or pancreatic amylase cannot cleave the cellulose. These enzymes are present in animals, but they cannot cleave the beta-1,4-glycosidic linkages present in cellulose. So cellulose is digested by an enzyme called cellulase enzyme. So this enzyme is absent in majority of animals. Some bacteria, protists and fungi produce cellulase enzyme. The greater animals, that is the higher animals, harbor these microorganisms in their gut for the digestion of cellulose. The microorganisms, they secrete cellulase enzyme and they help in digesting of cellulose for these higher animals. So thus a symbiotic relationship has been evolved between the cellulose eating animals and cellulose digesting animals. Now let us see about digestion of cellulose in termites. Termites are well known for their damage that they cause to wood and furniture. They voraciously feed on forest logs and other items which contain cellulose. The termites, they have flagellate protozoans called trichonympha in their hindgut. The wood they eat is not digested by this termite, but the trichonympha protozoan, it helps in the digestion of cellulose. It produces cellulase enzyme and digests the cellulose and releases the glucose. The glucose released is fermented by symbiotic bacteria to acetate. Nitrogen is also fixed to amino acids. The digested food is absorbed by the termite in the intestine. Apart from this trichonympha, termite also contains hundreds of species of symbiotic microbes. So these microbes and trichonympha, they will get shelter in the body of the termite, and termite will gain from these symbiotic microorganisms in digesting the cellulose. Now let us see about cellulose digestion in mammals. Herbivores have developed, that is herbivorous mammals have developed two adaptations for digesting cellulose. One is they have longer guts that provide more time for digestion and absorption of nutrients. And second is the fermentation chambers. They contain fermentation chambers which harbor microorganisms, and these microorganisms produce cellulase enzyme for the herbivorous mammals. The cellulose digestion is well developed in ruminants. The microorganisms which are seen in the fermentation chambers include cellulolytic bacteria, that is they break down the cellulose, hemicellulose and pectin. They are Ruminococcus flavifaciens, Ruminococcus albus, Bacteroides succinogens, Butyri fibrisolvens. And amylolytic bacteria include Bacteroides ruminocola, Bacteroides species, Bacteroides amylophilus, Selenomonas ruminantium, Streptococcus bovis, Succinomonas amylolytica. they act on sugar and starch. And protozoans, Epidenium echordatum, Dasytricha ruminaceum, Eudiplodinum maggi. Fungi include Aneromyces species and Sillamyces aberrancis. Now let us see about the digestion in ruminants. The word ruminate in Latin means to chew again. So the ruminants, they belong to the order Artiodactyla. The Artiodactyla includes goat, sheep, deer, cattle. So these animals, they chew the food twice. The food is regurgitated to the mouth and the food is chewed for the second time and re-swallowed. This process is called as rumination. In order to chew the food twice, their stomach is complex with 4 independent chambers. The chambers include one is rumen, the second is reticulum, third abomasum, and the fourth is abomasum. Truly speaking, this abomasum is the true stomach and the remaining 3 are projections of the esophagus. In the ruminant mouth, the food is mixed with large amounts of saliva and the food is partially chewed and converted into bolus. So that bolus is swallowed into the rumen, that is the first chamber of the stomach. In the rumen, the food is churned in the rumen with the help of the muscles and is partially, and the partial digestion occurs with the help of the microorganisms. The food is partially digested with the help of microorganisms. In the rumen, the food is churned thoroughly and the fermentation of the food starts. The plant material taken by the animal, the ruminant, it floats on the fluid. It floats on the fluid and this food enters into the next chamber called reticulum. So in the reticulum, the food is made into very soft bolus. So that bolus like structure is called as cud. So microorganisms are present in the reticulum and they digest the cellulose present in the food partially. And the food is made soft and such food is called as cud. So the cud is again taken into the mouth, is regurgitated into the mouth and it is swallowed, chewed and swallowed, and then that re-swallowed food enters into the omasum. In the omasum, the partially digested food and due to fermentation some products are formed. Those products are absorbed like water, vitamins and short chain fatty acids are absorbed. And later the food enters into the abomasum. Abomasum is the 4th chamber of the stomach and this is the true stomach. So in this abomasum, the lysozyme breaks the microorganisms so that the nutrients are released. And later on, the food enters into the small intestine. In the small intestine, the food is acted upon by the digestive enzymes and the food is digested and absorbed. The undigested food is adjusted out. Now let us see about mechanism of fermentation of cellulose by ruminant bacteria. The microbes present in the rumen and reticulum, they secrete a group of enzymes called cellulases. These cellulases are present on the cell surface, on the surface of the cell membrane in a structure called cellulosome. The cellulase enzyme will act on dietary polysaccharides, cellulose, hemicellulose, pectin and starch. So the hydrolytic species, they will convert these polysaccharides into soluble sugars first, that is the glucose. So then the glucose is again fermented with the help of microorganisms into short chain fatty acids like acetate, butyrate and propionate. So the glucose is oxidized by hydrolytic as well as fermentive species. It breaks them into first intermediary compounds like pyruvate, lactate, formate and succinate. Here hydrolytic as well as fermentative species, they both convert the soluble sugar into these compounds. And the fermentive species ferment and they make the end products as acetate, butyrate and propionate. So acetate is 2 carbon compound, propionate is 3 carbon compound and butyrate is 4 carbon compound. So here the methane and carbon dioxide is released during fermentation, and this methane gas, they are released during bleaching. Apart from digesting cellulose, microorganisms also supply nutrients like vitamins and amino acids required to the host. Now let us see about the role of gastrointestinal hormones on digestion. The digestive tract secretes a number of hormones that regulate the digestive secretions. All these hormones are peptides consisting of 4 to 43 amino acids. The endocrine cells are present scattered all over the gut, and they are not formed into a distinct gland. Some hormones are produced in the brain and are released at the target sites in the gut region. So the gastrointestinal hormones include gastrin, secretin, cholecystokinin, gastric inhibitory peptide, somatostatin, ghrelin, motilin and vasoactive intestinal peptide. Now let us see about each hormone in detail. The first hormone is gastrin. Gastrin is produced by the gastric mucosa, the G cells present in the gastric mucosa, that is the cells present in the mucosa layer of the stomach. Gastrin is secreted by the stimulation of the vagus nerve on taking a meal, distension and protein digestion. So it increases the HCl production, increases gastric motility. It contracts the lower esophageal sphincter and relaxes the pyloric and ileocecal sphincters. One of the most important functions of the gastrin include regulation of the gut mucosal growth. And the second hormone is secretin. Secretin is secreted by the enteroendocrine S cells present in the crypts of Lieberkühn of small intestine. Its secretion is stimulated by the acid chyme. Whenever the acid chyme enters into the small intestine, secretin is released. So the functions of the secretin include it stimulates the pancreas to release bicarbonate rich pancreatic juice and inhibits HCl production in stomach. The third hormone is cholecystokinin. This hormone is secreted by specific enteroendocrine cells upon the entry of acid chyme into the duodenum. The amino acids and fatty acids present in the acid chyme stimulate the cholecystokinin production. Cholecystokinin stimulates the pancreas to release pancreatic pancreatic juice which is rich in digestive enzymes, opening of the sphincter of OD and gallbladder contraction. Cholecystokinin inhibits the HCl production in the stomach, gastric motility, and reduces the hunger. The fourth hormone is the gastric inhibitory peptide, commonly called as GIP. This hormone is secreted by the enteroendocrine cells present in the small intestine. The release of this hormone is stimulated by the acid chyme with triglycerides, fatty acids, and glucose. The main function of this hormone is to inhibit the gastric motility and gastric juice production. The fifth hormone is somatostatin. This hormone is secreted by the enteroendocrine cells, D cells present in the gastric mucosa. This hormone represses the activity of growth hormone and also other gastrointestinal hormones. It inhibits the gastric secretion and its motility. It inhibits the intestinal absorption, pancreatic enzymes, and bicarbonate secretion. The next hormone is ghrelin. It is a recently discovered hormone. It is secreted by the A-type endocrine cells present in the oxyntic mucosa of stomach. Ghrelin plays an important role in the regulation of food intake, energy homeostasis, gastric emptying and acid secretion. It stimulates the appetite and causes intense hunger. Its secretion is more before intake and the secretion decreases when the stomach is full. Ghrelin is also known to stimulate the growth hormone. It also stimulates the growth hormone secretion. And the next hormone is motilin. Motilin is a peptide hormone. It is secreted by the mucosa of small intestine. It helps in the contraction of smooth muscles of the stomach and intestine. Motilin also stimulates acid and pepsin secretion by gastric mucosa. And the next hormone is vasoactive intestinal peptide. This hormone is secreted by the mucosa of intestine. Its secretion is activated by the entry of chyme. The secretion, the functions of the vasoactive intestinal peptide are release of water and electrolytes into intestine, inhibition of gastric secretion, and dilation of intestinal capillaries.